Okay, so I was just saying, after more exercise, that if it was me, I would just try to focus on the ones which are easiest to understand or interesting. So cloning cells in tissue culture is a very simple one that you could be aware of. You obviously need to replicate your DNA if you're going to clone cells. Your DNA profiling and fingerprinting you already need to learn about for forensics, so it's worth remembering that if you get a question about biotechnology and replicating DNA in the laboratory, and then you can mention your forensics and using the short tandem repeats, and the fact that if you are doing this DNA fingerprinting, you need to have enough of the DNA, so you might need to replicate the DNA so that you've got enough in the sample to compare it to other samples. So I would pick that one as well, and then maybe either your paternity testing, where you need to have enough DNA to find out if the child belongs to the father or to compare it to other men, and the gene splicing maybe. The gene splicing there might be a question on because it's very useful to produce these new things. I'm not so sure about the fishy tomato, but I'll leave that to you. So those are the 5 things which might come up in terms of replicating DNA outside of the body in the laboratory. It's the biotechnology. So that's the end of this Unit 1, part of Strand 1, before we go on to talking about meiosis. So what I wanted to do next was just to go through the gene splicing in a bit more detail for you. It is interesting, but it can be difficult to understand. And again, I will remake this in better light if you need to. So this is an example. We use E. coli a lot, as I've said, because it's a very simple genome. It's a bacteria, a single-celled organism. It's got a very simple genome, so it doesn't have a nucleus. The DNA or RNA is just in the cytoplasm, and we're able to map out all of the genes because there are very few of them compared with other organisms. And here we've got a gene from the pancreas, from a cell from the pancreas, which obviously produces insulin in humans and other mammals. And the idea is that we're going to remove this gene that codes for insulin production from this pancreatic cell. Okay, so we're going to cut it, basically, which means to splice that particular genome. And yes, Timmy is licking my hand. I don't know if you can see it. So we're going to cut it using enzymes. So we use an enzyme to splice the DNA at a certain point to remove the gene. We also need to cut the E. coli genome as well at a place where we're going to put this gene into the bacterial genome. So we need to splice the E. coli genome as well, which just means to cut it again using enzymes to make space to put in the one for insulin production. Okay, and then on the back we're going to insert the gene from the pancreas into the bacterial genome. Okay, so into the E. coli cell. And again, that involves using enzymes. One called ligase is used to stitch the genome back together. So it's like having to stitch it back together after you've cut it, basically. And then after that, you want that DNA to replicate itself so that you can produce more cells. Replicate the DNA to produce more cells. And each cell now has a gene to produce insulin. So you're going to get large quantities of insulin produced because you've got bacteria multiply very, very quickly, especially in the laboratory under controlled conditions. So if you've inserted a gene for insulin production, then you're going to have a huge quantity, a much bigger quantity than you could get through manufacturing it synthetically or from getting it from nature in any other way. So if we can get an organism that doesn't normally produce insulin to do so, then that's useful. And this process can also be used with growth hormone and other useful products as well. So I hope that made it a bit more simple. That's what gene splicing is about. So it's cutting up a genome and inserting another gene which will produce something useful, especially medicines that we could use. So I think that is it for today. You'll be pleased to know I do have a banana now. So in the next session I think I will try to show you how we can extract the DNA from a banana, and you can see a mass of it. But before I go too far, We'll start Meiosis next time. I do hope my bad technology isn't putting you off and you're still managing to benefit from this. But thank you for joining me once